So first of all, let me thank Alain for the kind invitation. So my talk will be divided in 3 parts. So first, I will briefly introduce the concept of London coding RNA and antisense transcription. Then I will share with you approaches and data that we have accumulated over the years on the presence of antisense and London coding RNA transcript in genes associated to hereditary Parkinson's disease and or selectively expressed in dopaminergic sostanza nigra. And in the remaining part of my talk, I will describe the identification or characterization of a London coding RNA that is antisense to UCLH1 and that actually is the representative member of a new functional class of London coding RNA that activates translation in a transcript-specific manner. So all of you know that since the sequence of a human genome, now the post-genomic era, thanks to the work of 2 large consortia. One hand ENCODE, the Encyclopedia of DNA Elements, which aim was to identify all functional elements in the human genome. And on the other hand, of a phantom consortia, functional annotation mRNA, that actually aim to assign a functional annotation to every transcript in every tissue and time in a mouse and human. Actually, we now have a complete different understanding of a functional characterization of a mammalian genome. And first and foremost, we now know that together, in addition to 25,000 genes that encode for proteins, we have even more number of genes, higher number of genes that actually does not encode for proteins. These are the so-called London coding RNA. You know, the last estimate actually points to 45,000 different London coding RNA transcripts in the human and mouse genome. On the other hand, now we also know that the majority of the genome is transcribed, and specifically in time and space. And this is specifically due to the regulated expression of repetitive elements of a long and of short type. So it has been postulated that, along evolution, particularly when we have an increase in heterogeneity of a cell type of a specific organism, we have a huge increase in number in London coding RNA, suggesting that London coding RNA regulated expression of heterotransposons are actually the major driver of evolution. So, but one of the most unexpected findings of this large genomic approach to transcriptomics has been the fact that almost all the genes in human and mouse genomes has a gene of the other filament. So the concept of antisense transcription. This has been, for the first time, analyzed at the genome-wide level in a paper from the Phantom Consortia, Katayama and collaborators, 2005 science paper, and then it's been, now proved through, in humans and even in bacteria, by Tom Gingera's lab in Cold Spring Harbor. So the realization that we have an antisense gene for every single gene in our genome actually leads to the definition of what is a sense and an antisense pair. So 2 genes share the same genomic region, but are transcribed for the 2 filaments. And if we look at them, how they could be reciprocal organized, actually we can classify them according to 3 different groups. In one hand, they are tail-to-tail convergent. In the other hand, they are head-to-head divergent. So in the first group, they overlap at the 3' end. In the other group, they overlap at the 5' end, or they can be fully overlapping, which one gene is completely contained within the other one. And we can also have all the different types of combination according to having protein coding genes that are antisense to protein coding genes, London coding RNA that are antisense to protein coding gene, and London coding RNA that are antisense to London coding RNA. Okay? And this is actually the configuration that has attracted more attention. This is not working, and in which you have a head-to-head divergent configuration, in which in one filament you have a protein coding gene, and the other filament you have a London coding RNA. And a lot of papers came out, in trying to understand how the London coding RNA actually is regulating protein gene expression. And we can now fairly say that every single, key regulatory step of gene expression can be regulated by an antisense. There are cases, for instance, in which antisense transcription actually is transcribing the promoter region of the same gene, and we know that the transcription factor can bind or cannot bind a transcription factor binding site in the promoter region of a protein coding gene according to the presence of antisense transcript. And clearly, also there are now an increasing number of cases in which antisense transcription is also involved in disease. As usual, first example came from thalassemia. So my laboratory is actually fully concerned in trying to address this fundamental question. Have long non-coding RNA antisense transcript any role in Parkinson's disease? And we approach this biological problem with two different approaches. On one hand, we look at antisense long non-coding RNA transcript associated to hereditary Parkinson's disease genes. On the other hand, we look at the same classes of transcript as expressed specifically in dopaminergic neurons. So, for both approaches, I just want to share with you a technology that now is present in many different variants, cage, deep cage, nano cage, helicos cage, whatever, many different types of variants and different applications. But at the crucial characteristic of a kappa analysis of gene expression case developed by Kaninichi, Yashizaki, and Riken is the fact that in the presence of mRNA, you copy a cDNA that forms a first-strand cDNA RNA duplex, and you isolate tags that correspond to the very first nucleotides of a transcript. Okay? So this tag is really a cDNA copy of the first nucleotides present in the messenger RNA. And then you sequence them massively. Then you get your bioinformatic data and you map it back to the genome. And what's happened, for instance, is that if this is a random part of a genome, in which you have that the height of a bar is directly proportional to the number of tags that actually map in this sequence, okay, you can see here that there is a huge number of tags that actually maps here and that are completely mapping at the 5' end of the ESTs that are known, transcribed for that region of the genome, in this case, calbindin. So what this means that tags from cage methods technology actually identify transcription start sites. Okay? And so you can do many different interesting things. For instance, here, Sanderling discovered that you can classify promoters according to the distribution of tags. You have very sharp promoters associated to Tata box, or you have very broad promoters associated to CPG island. Or you can look at alternative transcription start usage. And I think this is a crucial complexity in the transcriptome of mammalian cells that we overlooked. Look at this. This is a paper that we published in Genome Research some years ago, in which we did deep cage of different types of regions of the brain. And you can see here that you have of the very same gene, different transcription start site usage in different regions of the brain. And to these you can find a corresponding ESTs. So let's look at what we found in using this technology as antisense transcript in hereditary Parkinson's disease associated genes. So first of all, the most recent version of the Phantom project is FASTO5, in which they used cage technology, on a tremendous and huge amount of tissues. Okay, look at here. We are talking about 975. In the phase 1, 975 tissues in human and 399 different tissues in mouse, or samples in mouse. And this led to a publication in which we also were co-author in Nature last year. And the Phantom 5 cage actually takes advantage of a variant that is called helicos cage. But again, you produce capping, you produce tags at the 5' end, and then you sequence and you map. And what we did, we look at the distribution in all these tissues of the tags, in the loci associated to Parkinson's disease, hereditary Parkinson's disease genes. And look at what we found, for instance, in alpha-synuclein. So this is the alpha-synuclein locus. These are, the transcript for alpha-synuclein. You can see here the two alternative promoters that we all know are differentially used for alpha-synuclein. And here you can see on green antisense transcription. So this is the tags, the map in libraries from brain and cerebellum. Okay, so this is the transcription of alpha-synuclein gene. But these in green are actually transcription start sites associated to antisense gene. And actually, only here you can already see that there is an antisense transcript 5' head-to-head divergent long-non-coding RNA that maps at the 5' end of alpha-synuclein gene, and another one that actually is convergent in overlapping at the 3' end of, again, of alpha-synuclein transcript. This is just to give you, some example. So this is the two different transcription start sites of alpha-synuclein gene that goes in this direction, and these are transcripts that actually goes in the other direction. So there is a partially overlapping region, and these are the ESTs that are associated to these transcription start sites. But having this data for more than 1,000 tissue, we can also know exactly where these transcription start sites are used, how frequent and how high is the expression in that specific tissue. So we have a transcriptional map of transcription start site usage of all the antisense transcripts in alpha-synuclein locus. And this is, for instance, we always validated this transcript, and this is what's happening at the 3' end. So this is the transcription starts, these are different transcription start sites used in different tissue. For instance, you can see, we always found a very interesting pattern of expression in blood, okay, for all antisense transcripts associated to DJ1, alpha-synuclein, LRK2, and PINK, etc. I want just to, these are all the antisense genes that we found associated to alpha-synuclein locus. So you can see it's 1, 2, 3, 4, 5, 6, 7. So it will be very interesting now to understand how these 7 antisense transcripts that colocalize in alpha-synuclein locus actually are duplicated in the cases in which the gene alpha-synuclein is duplicated in some hereditary cases, actually modulate alpha-synuclein expression. And this is true also for DJ1. And here you can see some antisense transcript for DJ1, again validated by RT-PCR, and this is also for LRK2, in which we have a huge antisense transcription at 5' end that correspond to a head-to-head divergent long-non-coding RNA for LRK2. It would be interesting again to see how this LRK2 antisense long-non-coding RNA regulate parkate expression. Okay, but then we also study antisense transcription in all other genes involved in other hereditary neural disease, and this is just the map of a new antisense transcript that we identify, leading to a 12, we found 50 antisense transcription start sites. 12 were annotated long-non-coding RNA, 1 was annotated protein-coding gene in parking, and there was 37 new transcripts we are actually cloning by deep race and sequencing and characterizing. Okay, let me go back to the second part of my talk. All of you are familiar with the tyrosine hydroxylase, the antibodies that decorate dopaminergic neurons, soma, and dendrites in the mesencephalous mouse. And we resorted to, laser capture microdissection to try to identify genes differentially expressed between A9 and A10 neurons in the in the mesencephalous mouse. And in order to do that, we resorted to a variation of the cage that is called nanocage, that gives us 3 advantages. First of all, allow us to start from tiny amounts of RNA. It can work on poly A- and poly A+ RNA. Many of these long-non-coding RNA are poly A-, and in fixed tissue, partially degraded RNA. I don't want to go into the details, so we took advantage of transgenic mice with dopaminergic neurons who were labeled GFP. We took advantage of laser capture microdissection to differentially purify A9 and A10 dopaminergic neurons, and when we apply A9, when we apply nanocage on this part. Okay, I don't want to go into the details. This is just a general mapping of tags in the different, in RefSec gene, repetitive elements, non-coding RNA, intergenic regions. I want to say that 50% of tags are antisense to RefSec genes. And this is just to give you how, if you compare A9 and A10 pattern of transcription start site usage, you can see that in a random sequence of a genome, you can find transcription start sites that are used in both cells in the sense in antisense manner, but also transcription start sites that are selectively used in one of the two. So, by doing this approach, we have already published some work. One is that we discovered the expression of alpha-beta globin, alpha and beta chain of hemoglobin in dopaminergic neurons, particularly expressed in A9 dopaminergic cells. And also we recently published a paper in which we found that dopaminergic neurons express a repertory of olfactory receptors. Actually, the very same full-length cDNA sequence of some selected olfactory receptor that you find also in olfactory epithelia. And actually we de-orphanized some of them, and we show that carbon actually was able to trigger electrophysiological response in dopaminergic cells. And more recently, we just submitted a paper in which we show that dopaminergic neurons express a specific isoform of erythropoietin receptor. And this could have some interesting applications, in the use of erythropoietin as therapeutics for PD. But this is just to tell you the first characterization of long non-coding RNA expression, and by comparing long non-coding RNA expression data with olfactory epithelial nanocage data, we found 76 long non-coding RNA that are actually selectively expressed in 9 neurons and 55 that are expressed in 18 neurons, and additional 91 that are shared by the two. Now, by in situ hybridization, we have to validate this data and to see if, for instance, link RNA could be interesting candidates to look for subtypes of dopaminergic cells. But let's go a little bit better, more deeper in the characterization. One antisense long non-coding RNA. This is an antisense UCLH1 gene. Okay, so this is a classic head-to-head divergent sense-antisense pair in which we have a sense protein coding gene and the antisense non-coding RNA. I want just to point out that this is a 70 kilobase locus. The transcript is 2 kilobases. I want just to point out that the overlapping region, this is not in scale, is only of 73 nucleotides. And it involves the ATG of the UCLH1 transcript. So if you do laser capture microdissection on dopaminergic cells, you see that, that the antisense to UCLH1 actually is enriched in dopaminergic neurons. And then you confirm this by doing triple labeling experiments in which you, on the very same section, you look at tyrosine hydroxylase expression, and then you use 2 riboprobes for the sense of the antisense. As you can see here, the antisense, the sense transcript, the messenger RNA UCLH1, is present in the cytoplasm of dopaminergic neurons, and in the very same cell, there is also expressed the antisense, but the antisense is nuclear restricted. We will see later that this is an important piece of information. Okay, we recently published a paper in which we show the antisense UCLH1 is a component of a Nurr1-dependent dopaminergic gene network, and is now regulating neurochemical models of PD in vitro and in vivo. But what was more interesting for us is taking advantage, as I said, by Professor Perman, MA9, these cells that we received from very kindly from his lab. You know, that cells actually, that cell line actually express both sense and the antisense. So we clone the full length of the antisense, we transfect the antisense, and here you do real-time PCR to look at the transcript. The antisense is indeed transfected here, but has no effects on the RNA level of a sense gene. However, we then, by looking at the protein, we found out that actually the protein level was increased from 2 to 4 times according to the replica of the experiment. So suggesting that the antisense expression was indeed able to increase UCLH1 protein level with no effects on RNA level of a sense gene. All right, and since this work is already published, I don't want to go into the details, but what I want to share with you is the fact that this biological activity is due to the presence of 2 domains. On one hand, the 73 nucleotides that is the overlapping region between the sense and the antisense that we call the binding domain, and the inverted sign B2 element that is an embedded repetitive element. In inverted sign B2, there are 350,000 copies in the mouse genome. Some of them are not expressed, some are expressed independently as independent and transcriptional unit, but others are embedded in long non-coding RNA. Okay? And we found that actually, if we delete these 173 nucleotides of these inverted sign B2 elements, the long non-coding RNA of 2,000 nucleotides does not work any longer. Okay? So, is there any physiological condition where antisense UCLH1 regulates UCLH1 protein synthesis? I want just to make a long story short. We showed that rapamycin, so inhibitor of mTOR activity, actually increases UCLH1 protein level, and this increase requires the expression of the antisense. When we knock down the expression of the antisense or its function, rapamycin is not any longer able to increase UCLH1 protein level. Let me just summarize a very large amount of data because it has been already published. Okay, we know that there is cap-dependent translation going on in the cell, you know, and that this is possible because there is an assembly or initiation factor of translation, including the cap binding protein, that assemble with the 40 S subunits of the ribosome and the mRNA, and they start scanning for the cap, the 5' UTR, until a strong ATG sequence is found, and then the other subunit of the ribosome is associated and translation starts. So this is possible because in the cell, you know, there is TOR that is active. It's active by phosphorylating for ABP1, that is an inhibitor of the cap binding protein. In fact, when we inhibit mTOR, okay, by, for instance, pharmacologically by anti-rapamycin, 4-EBP1 becomes dephosphorylated, and this binds and sequesters the cap binding protein, AF4E, blocking cap-dependent translation. So I just want to point out that this is not found only pharmacologically, but is actually the first cellular response to stress. When cells is under stress, block cap-dependent translation, start choosing the mRNA that is going to be translated, and there's a component of the anti-stress response. And this is happening, in all these conditions. And in those conditions, what we know that some of the RNA are actually translated because they have a specific secondary structure in the 5' end. Okay, and these are called internal ribosomal entry site, or IRS, that does not need an active cap-binding protein. So what's happening in the case of antisense to UCLH1? What we have is that in growing condition, we have a UCLH1 that is actually associated to a light polysome, so it's translated, but not very efficiently, okay? The mTOR is active and is keeping cap-dependent translation going on, and the antisense to UCLH1 is nuclear retained, as I show you in vivo, in the section of the mesencephalon. However, when we treat cells with rapamycin, we block mTOR activity, we attenuate cap-dependent translation, and we trigger, we have several experimental data on this, some of them are published, we trigger the shuttling of the antisense from the nucleus to the cytoplasm. And through the overlapping region, the antisense triggers the shifting to the heavy polysome of the mRNA through UCLH1. So in the presence of the antisense, the mRNA UCLH1 shift from light polysome to heavy polysome for an increased rate of translation. And this actually is responsible for the increased UCLH1 level that we see in our Western blot analysis. Okay, so since then, what we in testing this model, the first thing that we did actually was by looking at different RNA-seq, helicose cage, and small RNA, the type of RNA that shuttled from the nucleus to the cytoplasm upon rapamycin. And we found, several differentially expressed genes in the cytoplasm upon rapamycin. And what was interesting that an important fraction of the cells of genes that shuttle actually are components of the sense and the antisense of the very same gene, okay? In the very same sense-antisense pair. And we are actually characterizing now what is the significance of this, and this is the base of a molecular mechanism. But what I want also to share with you is the fact that we ask ourselves if this is a characteristic only of the antisense H1, or is it a general characteristic shared by other natural sense-antisense pair. So we look bioinformatically and we search for sense-antisense pair in which sense is protein coding, they are 5' head-to-head divergent, and the antisense London coding contain the overlapping region and an inverted SINB2 element of a very same type of antisense H1. And actually we found 200 sense-antisense pair, and this is just one example. We have now validated more than 10 of them, in which we resorted to study a sense-antisense pair that is expressing M9D dopaminergic cells. We clone the full length, we transfer the full length of the antisense, and again, no change in UCLH1 in UXT mRNA level, but increasing protein level. And so we decided to call this class of natural antisense long-encoding RNA SINEUP, because through the activity of a SINE element, they upregulate translation. So, what is the embedded domain acting in human? Because SINB2 elements are not present in human genome, okay? So this is a very interesting aspect of studying repetitive elements in the evolution of genomes, because different types of repetitive elements have parasitized the genome in different species, playing a fundamental role in evolution. So we ask ourselves, are there any type of repetitive elements that can functionally rescue the activity of SINB2 elements in human? And actually it's yes, and actually we found it in collaboration with Piero Carnici, finding that a sense-antisense pair of a human protein phosphatase 1 regulatory subunit to a locus, in which we actually found that through the activity of embedded repetitive elements, actually we have this antisense is working exactly as the antisense UCLH1 in mouse. But then we ask ourselves how the effector domain is working. What is the functional mechanism? What is the molecular mechanism of inverted sign B2 elements to activate translation? So you know that inverted sign B2 elements has a pol 2 DNA binding site, as a B box and A box that is involved in RNA pol 3 transcription. And so we resorted to chemical footprinting in order to study the secondary structure of inverted sign B2 element transcribed in vitro. And this is actually the results in which, you know, this is the 5' end. So this is the structure in which it forms 3 different stem loops and 4 different internal loops. And then we tried to make, we deleted all different sequences in the structure, and we found out that by deleting selectively the very top 5' end, the very top end of this secondary structure, that is the stem loops, we can kill completely the activity. So, okay, so this is not trivial. You know, you have a long non-coding RNA of 2,000 nucleotides. You delete 9 nucleotides, and you know, according to the structure that you identified, as is not working any longer. So we resorted to NMR studies of a 38 mel of a stem loop structure in the inverted sign B2 elements, and we found out that actually it's forming, you know, an A-type RNA helix, and then a pseudo loop at the very top that is formed GUG3 loop and 2 highly unstable AU base pair. And then we did the molecular dynamic simulation. Here you can see here that this is actually the stem, and this is the pentaloop structure. Okay, and we are now better characterizing the presence of the the functional significance of this stem pentaloop, because it's very, as you can imagine, it's very similar to a tRNA. So one fundamental aspect in finding out the mechanism is to try to look at the presence of or identify proteins that binds the RNA, and we resorted to a screening of phage display library, and actually we screen this off library with different probes transcribed in vitro, and the major interactors actually resulted to be this double-strand RNA binding protein interleukin enhancer binding factor 3, and we have recently, 2 weeks ago, actually validating the interaction between IL-3 and the protein, and what is interesting, and the antisense USLH1, and what is interesting here, that actually IL-3 is very well known to shuttle from the nucleus to the cytoplasm according to rapamycin treatment. Actually, we recapitulated this phenomenon in M90 cells, and we are actually now characterizing the RNA interactome on IL-3 to show if IL-3 is indeed, you know, a fundamental molecular player in nuclear cytoplasmic shuttling of synapse. Right, so let's go back to the concept of synapse. So this is delta 5' synapse. So it's a synapse with only the effector domain. But if the binding domain so that is the pairing is responsible for choosing the type of mRNA that must be translated, then we can, you know, modify that sequence. And for instance, you know, we, as a proof of principle, we created an artificial binding domain to GFP mRNA, proving that actually it was working as expected. That is, you know, if we translated increasing amount of the synapse GFP, you know, no change in RNA level for GFP, but increasing GFP protein level. And this is actually can be complemented also by if we put the repetitive human repetitive elements that we found in PP1R112A. So the human repetitive element is able to complement in a synapse GFP the activity of inverted sign B2 element. Okay? But then we can design, so synapse actually works in different cell types. Here there are some examples, HEC, HEPG2, HeLa, we know now also keratinocytes, neuro2A, etc. And then we look at synthesizing different synthetic synapse for different targets. This is a synapse flag, in which you can see here, you design a synapse flag, and you have a flag, a fusion protein to a gene, to a protein that we study in the lab. It's called TRAF6. As you can see here, if you co-transfect flag TRAF6 and the synapse against the flag, you see increase of a fused protein by an anti-flag antibody or an anti-TRAF6 antibody. On the other hand, we also synthesize synapse against secreted proteins. There are some natural synapse that are for secreted proteins. For instance, one is an antisense to elastin that actually works. We proved, and we just submitted a paper, work as a natural synapse. But also we designed synthetic synapse to increase production of recombinant antibody, clearly to be, for a potential protein manufacturing application. So, but if we want, so now we ask ourselves, could we design synthetic synapse to increase the level of a target gene protein, at will? And in order to understand, in particular, this could be a huge number of applications, in molecular therapy. These are all the clinical trials, they use small RNA as reagent and drugs, but you all know that these are all inhibitory. These are all antisense oligonucleotides of siRNA molecules. So none of these RNA actually able to increase the expression of your target. So in order to prove that, the first thing that we need was to create a mini synapse. So not using a 2 kb transcript, but go down to 246 just to start. This is just obtained by putting together the effector domain and the binding domain, only those two sequences. And now we are chopping the effector domain, and we know that binding domain can work as short as 32 nucleotides so far. So then, but the fundamental question is, are synthetic synapses able to increase endogenous protein level of genes that do not present a natural synapse? And so, since we work in Parkinson's disease, we were interested in developing synapse for DJ1, and we created a synthetic synapse for DJ1. As you can see here, that by transfection with a synthetic synapse for DJ1, we are able to increase DJ1 protein level of 3-fold with no change in RNA level. But the most interesting application could be, haploinsufficiency. All you knows what are they. They occurs when a diploid organism is only a single functional copy of a gene, and the singular functional copy does not produce enough protein to maintain wild-type condition, leading to an abnormal or disease state. Synapse could be an important tool because, first of all, they act on a specific mRNA isoform. Second, there is no uncontrolled ectopic expression. For those of you that works on transcription factors, know that if you overexpress 50-fold the transcription factor, you have a lot of, a low specificity binding sites that are activated and a lot of specific effects. And the increase here is in physiological level. So we created synthetic synapse against, for instance, frataxin, for Friedreich ataxia, and here you can see a synapse for frataxin that actually is able to increase endogenous frataxin level 2.5-fold. But then we also, and right now we have an active collaboration with 11 laboratories worldwide for 14 haploinsufficiency, 20 target genes, and we have several positive results. I want to say that my major effort right now is to create synapse for the different isoforms of BDNF mRNA. Because the BDNF has one exon for a pyridine frame, but has 8 alternative 5' UTR with 8 alternative promoters. And if you can increase the translation of BDNF only in one specific isoform, then you have been much more selected in your activity. So let me just share with you in the last 5 minutes of my talk, results from collaborative laboratories. This is Giuseppina Miano, Igib in Naples, that designed a synapse against KDM5C, a lysin-specific demethylase that gives rise in haploinsufficiency and X-linked intellectual disability. And you can see here that actually synthetic synapses are able to increase endogenous endogenous KDM5 protein level, both in two different cell lines, and are actually able also to rescue the function of the protein that, so in demethylating targets that were previously methylating for the low expression of a protein. But most important, most interesting, we were very fascinated in trying to see if synapse can work also in non-mammalian chordates. It could be a reagent like morpholinos, okay? And so we collaborated with Brunella Franco at TGEM, and we designed synthetic synapse against different genes involving microphthalmia with skin lesions that have been modeled by Brunella Franco in Medaka, okay, in fish. So this is a test of a phenotype in vivo in a fish by creating synthetic synapse. I don't have time to go into the detailed experiments, but you can see here. So when you have a morpholino, you see a degeneration, basically a undeveloped eye. But if you do the morpholino plus the synapse, you have basically the rescue of a phenotype. And this has no effect on the level of a target RNA. So we have several proofs that this is due purely post-translational. We are now, so this could suggest that we can create a library of synapse for specific mRNA target to study in vivo, in Medaka and perhaps also in SINEUPs, etc., as for morphogene or for knockdown genes. So synapse at the end could be used as experimental tools in the lab for biotechnology reproduction, and we hope in the future for therapeutics. But let me just finish with what is my take-home message. Okay, when one talks about long non-coding RNA, probably has a misconception. The long non-coding RNA are linear, single sequence, unique sequence transcript. Indeed, 40% of the nucleotides that are present in a long non-coding RNA are embedded repetitive elements. This is important, because if you start thinking about that, and we can take advantage of the data that I just presented, we can now hypothesize a molecular domain organization of long non-coding RNA, in which embedded repetitive elements are actually the functional domain. And through a repertory of RNA protein interaction of the different embedded repetitive elements, we can then tether a specific biochemical activity or subcellular localization to a specific RNA target to mRNA-RNA interaction, or to a specific genomic region to DNA-RNA interaction. And so let me just finish by thanking my collaborator, in particular Piero Cannici at RICEN and Claudio Santoro at University of Eastern Piedmont in Italy, and my lab, in particular Silvia Zucchelli and Claudia Carrieri. Thank you.